Hello, my name is Ethan Blecker, and today we'll be talking about transcription, protein synthesis, and the Lac-Operon process. In transcription, the transcription factor sits on DNA. The RNA polymerase then strands from 3 to 5 template. It starts to copy the lower template into RNA. The RNA polymerase continues to move along DNA to make the RNA copy, which is then released. The 5 cap is then added and adds guanine to the 5 end. Poly A polymerase enzyme adds adenine nucleotides to 3 end of RNA. The spliceosome RNA protein complex removes intron and then reforms it into a loop, which is then cut, and the RNA econs rejoin, forming a complete RNA. Initiation is the first stage of protein synthesis. The start codon has small subunits of mRNA that bind to it, the complementary base pairings in this sequence. The next stage is elongation. This is a series of repeated cycles that add amino acids one at a time to make a peptide. The tRNA and amino acids match the start codon and slot onto the mRNA. The amino acids then overlap, which is the start of a peptide bond formation. The ribosomes move along mRNA by one codon. Peptide bonds formed between amino acids gets bigger. Termination is then the last stage, when the peptide bond hits a stop codon, and this then stops the protein synthesis, and a peptide bond is made. The repressor protein is always present and is active when lactose is not present. Repressor binds to the operator, forming a loop. The repressor stops the DNA polymerase from transcribing the Lac-Operon genes in the absence of lactose. The repressor is always made, but is not active in the presence of lactose. When lactose is present, the RNA polymerase is able to transcribe the Lac-Operon genes. A single polysomic mRNA is produced. RNA polymerase transcribes the Lac-Operon gene so the bacteria can use the lactose.